RAA Technical Introduction and Experimental Operation Demonstration. RAA technology is the abbreviation of recombinase-mediated isothermal nucleic acid amplification technology and is a patented molecular diagnostic technology independently developed by China. The RAA technique is a technique for performing nucleic acid amplification under isothermal conditions, optimal temperature 37 degrees Celsius, using a recombinases, a single-stranded binding protein, and a DNA polymerase. The principle of RAA is as follows. Recombinases forms a complex with a primer, scans the double strands, invades the double strand, and opens the double strand. Then the single-stranded binding protein keeps another strand in a single-stranded state. Polymerase is then responsible for extension and amplification in the presence of dNTP. Repeating this process, the amplification signal will be detected by a portable instrument in 5 to 15 minutes. Compared with traditional PCR technology, RAA technology has the advantages of isothermal, rapid, portable, sensitive, specific assay, cheap equipment, and simple operation. Currently, RAA has been applied in the fields of inspection and quarantine, disease prevention and control, food safety, species identification, and environmental microbial detection. It is particularly suitable for POCT, disease early screening of acute infectious disease. The commercial RAA detection kits include, but not limited to, human respiratory syncytial virus, adenoviruses 3 and 7, hepatitis B virus, Bordetella pertussis, Mycobacterium tuberculosis, Schustason, and African swine fever virus. The following is a demonstration of the RAA experiment. The items required for the experiment include nucleic acid sample, nucleic acid detection kit, centrifuge tube rack, PCR tube rack, pipette, pipette tips, and waste tank. The nucleic acid detection kit provides reaction buffer, magnesium acetate, negative quality control, positive quality control, enzyme reaction unit. Experimental settings were negative control, nucleic acid sample to be tested, positive control. The instrument is first warmed up before the start of the experiment. Open the computer software of the fluorescence detector. Select the serial port number corresponding to the instrument and connect the communication. The enzyme reaction unit is taken out and marked on the tube cover. And the reaction buffer, negative quality control, positive quality control are thought for use. A reaction buffer was sequentially added to each enzyme reaction unit. Magnesium acetate was added to the tube cover of each enzyme reaction unit. A negative quality control was added to the negative control tube. A nucleic acid sample was added to the experimental tube, When loading, it is necessary to add samples according to the sample concentration from low to high. When the sample is added, the lid of the reaction unit needs to be buckled to prevent cross-contamination, and a positive quality control was added to the positive control tube. Put the reaction unit symmetrically into the oscillating mixer, and click the pre-treatment button to mix and centrifuge. Place the mixed reagent into the fluorescence detector, and click the Run button to perform the fluorescence detection. Real-time amplification curves as shown in the video. Amplification results can be observed in 5 to 15 minutes. After the end of the run, set the slope parameter according to the kit instructions, and the software will display the analysis result on the left side. The above is a demonstration of the experimental operation of the fluorescent Raa nucleic acid detection kit. Raa technology can also be applied to gel electrophoresis, test strips, reverse hybridization chips, and other detection methods. Thanks for your watching.